Welcome to Unit 2.2. This is our Introduction to Genetics and our talk about mutation. If you need a little bit of review, I posted a video in this same folder that talks about DNA and about the qualities of DNA and a little bit about the overall outcome fitness-wise of mutation. So that's a good place for you to look at and review that. So genetics was one of the large mysteries in biology for many, many years. People understood that traits were passed from one generation to the next, but the mechanism of how that occurred was, for a long time, completely unknown. And so we talked about in last unit how at Darwin's time most people accepted this idea of a blended inheritance, which is the idea that once a mother and a father came together to make an offspring, that offspring was half of each parent. And this made a lot of sense based on phenotypes and that offspring do look a little bit like one parent and a little bit like another parent. But the idea was that it was something unique and completely mixed together. A model that is often used to make an analogy for this is like mixing paint. And it wasn't until Mendel came around with his very careful, meticulous experiments on pea plants that he began to understand that a particulate method of inheritance was actually the correct one, and that is what we understand today, that we have individual genes, that most organisms that we're studying are diploid, meaning they carry two copies of each of their genes, and those two copies do not fuse together to make a third unique structure. They remain separate and independent. So if you have, for instance, A blood type, you could either have a big A allele from your parent, which is dominant, a big A allele from your other parent, say the mother, which is dominant, and you would be homozygous and be of the A blood type. You could also have an O allele from one parent and a big A allele from the other parent, and that would create a blood type also, because O is completely recessive to that A blood type. And so this idea of genetics really began to be understood once this particulate inheritance was fully understood. And Darwin lived in a time when this was still something of a mystery. Now, once Mendel figured this out in the mid-1800s, he published his work. It was in German, in a German language journal, in a journal that wasn't very well, wasn't a very large, important journal, and his work largely was ignored. People didn't realize the breakthrough that he had come, and it took almost 80 years before people rediscovered his work and began testing it and finding out that it worked not only in pea plants, but in fruit flies, in humans, in all animals. Okay, so a couple of vocabulary words you need to review. Meiosis, which is of course a form of cellular division. It's a cellular division in which the genetic material is divided in half, and the end result of meiosis are gametes. So we make eggs and sperm. And gametes, of course, are a cell carrying half of the genetic material that are able to fuse with another cell to form a organism. That's called fertilization. Okay, again, you should know this already, but a bit of a review or a refresher if you don't. Locus is a technical term for a location on a chromosome. It's an address, if you will. So we could look at chromosome 1 and look at a particular position on that chromosome. We would call that a locus. The plural of locus is loci, L-O-C-I. So just be aware of that term. You might see it come up now and then. The law of segregation is the fact that traits tend to segregate out independently and are not dependent on one another. So let me give you an example. If we were to, say, select artificially for a certain trait in a population, let's say we wanted dogs that were larger than average, and so we would choose the largest dogs in populations, breed them together. On average, their offspring would be larger, and if we continue that process, we could over time get larger and larger dogs. And that's what's happened in many of our domesticated breeds. Great Danes have come from that process where they've chose larger and larger dogs and bred them together. Now, just because you choose for size doesn't mean that you're necessarily choosing for any other factor. So coat color. Large dogs and coat color, you can have Great Danes of all different colors, and by selecting just for large size, you're not picking necessarily for any other trait. So that's the law of segregation. And finally, in genetics, it's very common to have dominant and recessive genes. Mendel was studying dominant and recessive genes, but there are lots of other combinations that can be a little bit more complicated, and We're not going to spend a lot of time in this. That's really something more for the province of genetics class, but you should understand the basics of genetics and that some traits are dominant over others. Now, be careful as we are talking. Remember that just because a trait is dominant or a trait is recessive, it's not saying anything about its fitness, whether it's a good or a bad thing. You can have recessive traits that are very fit, traits that are very adaptive and allow organisms to do very well in their environment, and you can have dominant traits that are adaptive and allow organisms to do well in their environment. So what's good or bad does not depend on whether it's dominant or recessive. Okay, and we'll talk about the law of independent assortment, which is more commonly known as sometimes is known as random assortment. We'll talk about independent assortment of alleles. Okay, so you should also know DNA. That's a nice easy one from Bio 1. And then in most eukaryotic organisms, the DNA is divided into individual pieces. And these are called chromosomes. And these can be very, very large, and there can be many of them. So on the bottom left, we've got a table here that shows the number of chromosomes in many different eukaryotic organisms. So humans are down here in the bottom right. We have 46 chromosomes. Commonly, that's listed as 23 pairs of chromosomes. You get a chromosome from mom and a chromosome from dad, and then that is repeated 23 times. So each gamete, human gamete, carries 23 chromosomes. It fuses with another gamete, has another 23 chromosomes that match the 23 in the first gamete, and those form the total 46 chromosomal count of human beings. Now, during meiosis, this process of independent assortment creates all sorts of combinations and varieties. So I've got this question listed here on the bottom right. What are the odds that you're genetically identical to a sibling? Now, be aware we're talking about siblings that are share the same parents, so the same mom, same dad, but are not identical twins. In identical twins, these processes we're going to talk about here all happen prior to the embryo splitting into two individual embryos, and so genetically those two individuals are identical. Now I've got a little picture here. This was a drawing of DNA, a very kind of cartoony diagrammatic version of what DNA looks like, but review that, just make sure that makes sense to you. If not, you can go back and look at the other video that I posted in this folder, which gives you a little bit of an overview of DNA, but it's something you should have picked up previous to this class. Okay, so let's think about this question. The odds that you are genetically identical to a sibling who is not your twin. So, during meiosis, chromosomes find their pair. So we've got a drawing here in the top left of a cell that has two different chromosomes. Chromosome number 1 is listed on the top, and there's a blue one and a red one. So the blue one came from dad, the red one came from mom. And prior to meiosis, those chromosomes are duplicated, and then they find their match and they line up. So here we have on the left, blue chromosome from dad finding its match. This might be chromosome 1A, and then we find chromosome 1B from the mother, and they line up during meiosis. The second pair of chromosomes also finds its match and lines up. However, there is no mechanism to make the dad's chromosomes all line up on one side or vice versa, and so they line up randomly, or as the more common term is, independently in reference to the other chromosomes. So you can get all sorts of different combinations. And so a good analogy for this is, think of it like flipping a coin. When a gamete is formed, it's basically flipping a coin, whether it's chromosome 1A or 1B that gets incorporated into that gamete. And so when we flip a coin, every time we do that, it's like a 50-50 chance. Okay? So because most eukaryotes have more than one chromosome, this makes a massive amount of combinations. An analogy for this is like if you flipped a coin twice. If you have two pairs of chromosomes, it would be like flipping a coin twice, recording what you got. So let's say you got heads on the first flip and then tails on the second flip. So then you would say, what are the odds that I repeat that series? That if I flip a coin twice again, I would get heads on the first flip and tails on the second flip. And the odds for this, the probability is pretty straightforward. So for the first coin flip, it's a 1 in 2 odds that you would get a heads, so basically 50-50. And then the odds that you would get the same thing on the second time is also 50-50, but the odds that you would get both of those things in that series with heads first and tails second is 1/2 times 1/2, or 1 out of 4. So only 1 out of every 4 coin flips is going to be heads, tails. Another way to think of this is to think about all the different possibilities. You could get heads, heads. You could get heads, tails, which is what we were actually trying to repeat. Or you could get tails, tails, or you could get tails, heads. And so those 4 possibilities mean you have a 1 in 4 chance of getting any one specific series. So if we extend this beyond just 2 coin flips, then we can mathematically display that as this equation here, 1 over 2 to the n power, where n is the number of coin flips. So 1 coin flip, 1 out of 2 to the first power, which is 1 out of 2. 2 coin flips, 1 out of 2 to the second power, which is 1 out of 4, and so on. Now, since humans have 23 chromosomes, every time one of those chromosomes independently assorts, it's again like flipping a coin, like 50-50. And so with 23 pairs of chromosomes, we have a 1 out of 2 to the 23rd power chance of having the same series of assortments when we're assorting the chromosomes. And if we do that math and extend that, it's a little bit over 1 out of 8 million. So when mom is going through meiosis to make the egg that made you, there are a little bit more than 8 million different possible combinations of chromosome rearrangements. So the chances that you get the same sets of chromosome from your mom that your sibling got is 1 out of a little bit more than 8 million. Now, if we're going to extend this and say, okay, that's the chance that the egg that was sorted, the chromosomes that were sorted into the egg that made you is the same as in a sibling, is 1 to 8 million. That has to happen exactly the same in the father. So in reality, if we're looking at both the egg and the sperm getting together in that same extremely rare combination, we times 8,388,000 times 8,388,000. And so there's a 1 in a 70 trillion, yes, that's trillion with a T, 1 in 70 trillion chance of having 2 siblings be genetically identical because of this process of random assortment. So the take-home message is that random assortment creates massive amounts of genetic diversity, and it's nothing that wasn't there before, it's just mixing and matching the diversity that's already there. So it's maybe a different way or a different form of diversity. So more genetic definitions. An allele we've talked about already, that's a version of a gene. A common example is blood type. There are 3 different alleles for blood type in the human species. You can have A, B, or an O, and then you get 1 of each from parents. So if you get an A from dad and a B from mom, you have an AB blood type. If you get a B from dad and an O from mom, because O is recessive to B, you have a B blood type. And if you get 2 O alleles, then you have the O blood type. Okay? So if you get 2 alleles that are identical, that are the same, you are a homozygote for that gene. If you have 2 different alleles, you're a heterozygote. These should be familiar terms for you. The genotype, of course, is all of your genetic tendencies and those alleles combinations, and then your phenotype is physically the way you look. And we've talked about how your phenotype is predicted by both your genes, nature, and also by your environment, which is sometimes called nurture. So this is the old nature versus nurture. And so phenotype for some traits is mostly determined by your genes and only a little bit by environment. For other traits, it's vice versa. And for some traits like heart disease, for instance, it's a combination of both your genetic background and your environment, maybe even roughly equally. Okay? Now, there are other parts of the genes called regulatory elements. These are places that are actually nearby a gene and help to control the expression of that gene. So it's going to control whether or not a gene is turned on, meaning is it transcribed and translated and used in that cell, or is it turned off, meaning is that DNA just kind of wrapped up and packed away so the gene is never used. And so regulatory elements can be critical for phenotype also. It's not just the alleles you have, it's how those alleles are turned on and used. Okay? Now, there's actually one additional source of variation during meiosis, and this is crossing over. So when chromosomes are paired up during meiosis 1, this is metaphase in meiosis 1, they can swap parts. So my analogy is, imagine that chromosomes are like a dance partner. And in meiosis, you find your dance partner and line up along the center of the room or the cell if you're looking at chromosomes. And those partners then will separate as the cell divides. But imagine you looked over at your partner and said, hey, you've got a really nice arm. I like your arm. And your partner says, oh, I like your arm. And said, well, why don't we swap? And so you would swap arms. Now, hopefully you've never been to a dance like that. It's kind of a little freaky, but that would mean that one of these chromosomes is mostly inherited from, say, the dad of that individual that's going through meiosis, but a little part of it would be from the mother. They would have a Frankenstein, so to speak, like a Franken chromosome. It's a little bit of one and a lot of another one. And so we end up here with a chromosome that's mostly from the dad in blue, but a little bit of mom left over. So remember back here where I told you that the chances of you being genetically identical to a sibling are 1 in 70 trillion? It's actually much greater than that, because not only do we have the variability that can be created by independent assortment, but we also have additional variability that can be created through the process of crossing over. So you have a more than 1 in 70 trillion chance of being genetically identical to a full sibling. So there are massive and massive amounts of combinations that can be made just simply through the process of sexual reproduction. Now, when we look at DNA, we need to be able to understand and remember how we go from the genetic code to the actual expression of the phenotype. In a way, this is like the language of life, right? How we get from the DNA molecules to some useful protein product. Okay. Now, to do this, we are going to look at these individual base pairs. Now, by the way, sometimes there are several different words that are used for the individual repeating units of DNA. When they are paired together, they're most often called base pairs. So A, adenine, pairs with T, thymine, and we call that together a base pair. Now, we only need to look at one side of the DNA to know what the other side is, because it's always complementary. So another word that's sometimes used is a nucleic acid, or maybe more commonly a nucleotide. And a nucleotide is just one component of a base pair. Most frequently, we use the nucleotides that are in the coding sequence that will then be transcribed and translated into a gene. Okay, so vocabulary words, review from Bio 1 again. Transcription is the process of taking DNA, a gene, and transcribing it into RNA. And because DNA and RNA are the same type of molecule, they're very similar, we call that process transcription. It's as if you took something in English and made a copy of it also in English, or at least in a very, very similar form of language, maybe English in a different dialect or something like that. But you basically have a copy of the same thing. So that's taking DNA and getting a copy of it in RNA. That RNA, if it's an mRNA, is then taken to a ribosome, and it is translated into a amino acid. And because amino acids are chemically quite different from nucleic acids, it's a very different type of a molecule, we call that process translation. So an analogy would be like taking English and translating it into Spanish. The information is essentially the same, but it's an entirely different form. Okay, so that's the process. So know an amino acid. These are the basic building blocks of proteins. So nucleotides string together and make nucleic acids, DNA and RNA. Amino acids string together and make proteins. And we'll look at that here in a little bit. Okay. Now, how we go from a nucleic acid to amino acid, this translation process, the individual fundamental unit of that is called a codon. And a codon is a series of nucleotides that together can then code for a single amino acid, and that's why we call it a codon. So at the bottom I've got this question: how many codons do we need to code for all of the amino acids? Well, here we have a chart showing you the translation of the genetic language into the amino acids. And so the way that you read this, and you'll need to be familiar with this type of a genetic chart, we're going to be using them for worksheets, and you'll see them again in various parts of the lecture. This inner circle here represents the first position of a codon. Okay? The next outer, I guess the middle ring here, represents the second position of the codon. And this outer ring of letters, A, C's, G's, and T's, or in this case U because it's in RNA, that represents the third position of a codon. Okay? And so we have to have 3 codons to code for all of the amino acids. To understand why 3 is the so-called magic number, let's take a look at the variety and diversity that we see in amino acids. Okay, so first, how many different amino acids are there? Well, it turns out that there are 20 common amino acids. These are used in nearly all proteins and are the essential building blocks for proteins. There are a couple of rare ones that are used in very, very extreme but very limited situations in some organisms, but for the vast majority of organisms, they only use these 20 amino acids. Okay, so we need to code for at least 20 amino acids. Now, if each of our codons was only one nucleotide long, we could only code for 4 different amino acids, right? An A could represent one amino acid, a C would be translated into another amino acid, and so on down the line. So 4 is not enough. We need to do 20. If we had 2 nucleotides, then now we have 16 different possible combinations, AA, AC, and all the combinations that you see listed here. I actually left out, sorry, there's a whole string here that could be possible. I've only listed 12 of them here. But you have 16, so you can, in addition to these 4, have, these 12, you could have TT, TA, TC, and TG. So we have 16 possible that that you could have if you had 2 nucleotides and a codon. So still not quite enough. We need 20 amino acids. We'd only have 16 codons with 2 nucleotides. So finally, once we get up to 3 nucleotides, now we can have enough codons to code for each amino acid. So there are 64 possible different combinations if we have 3 nucleotides in our codon. And so that's the right answer. There are 3 nucleotides in each codon. But notice something here. We only need to code for 20 amino acids. However, we have 64 possible codons that are available to us. And so this results in something that we call degeneracy. And degeneracy is a little bit of a funny word. When we think of degenerate, which is a similar word to degeneracy, we think of something that's degraded or has become less than what it was before. It degenerates. So degeneracy is maybe not the best choice of words, but it's the correct technical term for it. A better word, when you think of degenerate, think of redundant. So basically there is redundancy in the genetic code. So for instance, if we looked at one of these codons, CCC, let's go back here to our genetic code. So notice that C, first position, C, second position, C, third position, codes for proline. However, if we had a CCA or a CCG or a CCU, those would all also code for proline. So that's the redundancy, again technically termed degeneracy, of the genetic code. So we have 64 possible codons, but we only have 20 amino acids. Now, some of the codons are used up as stop codons, and so that takes away 3 of them, but that still leaves 61 different codons for only 20 amino acids. The start codon, AUG, also codes for methionine. So methionine is the first amino acid in all proteins, and it can also be an amino acid in the middle or near the end of the protein. So AUG is both a start codon and the codon for methionine. Okay? Occasionally, during DNA replication, mistakes are made. The most common type of mutation, the mistakes that are made during DNA replication, is a point mutation. And so we're going to be mainly talking about point mutations and their impact on organisms. We'll talk generally about some other major large mutations that can occur that are less common but do occur also. So let's look at point mutations and let's define them. There are 2 major classes of point mutations, and then within this first one, substitution, we're going to define different subclasses. Okay? So a point mutation, very simply put, is when we have a change, a difference of 1 nucleic acid. And a substitution is where 1 nucleic acid is swapped out, a mistake is made, and a different amino acid is put in there. So if we're reading the original sequence, it might say CCC, but a mutation might change that to a CCA. So it's a real difference. It's carried in the genome of the organism that inherits that mutation. And again, the ones that we are most interested in for evolution are mutations that occur during DNA replication, during meiosis, if we're looking at sexually reproducing populations, or just regular replication for mitosis if we're looking at an asexual organism like an amoeba or a bacteria, because those can be passed down to their daughter cells. But in eukaryotes, it's only the mutations that occur during meiosis that can be passed on to the next generation. And so those are the ones that we're very interested in and that we are concerned with when we're talking about evolution of populations. Okay, so let's define different subclasses of substitution. These are ones that you need to know. The first class of a mutation is a synonymous or a silent mutation. And those words are used often interchangeably. There's a slight difference. A synonymous difference is a change in a coding sequence that leads to no difference at all in the amino acid. So let's go back to our example. If we had a CCC in the original genome of a parent, and when that parent was copying their DNA to make gametes, a mistake was made, and instead of CCC we had a CCA. We would still be coding for proline. Even though that's a real mutation that the offspring would carry, it phenotypically would make no difference at all in that offspring, because you would still, when you transcribed and translated that codon, you would still make a proline, and the outcome, the amino acid, would be the same as it was before the mutation. So that is a synonymous mutation. Now, if a mutation occurs outside of the coding region of the DNA, so in the non-coding DNA, it can also be silent. It doesn't change a codon because we're not in the coding region anymore, but it can be silent. So technically, a silent mutation is any mutation that does not impact the phenotype of an organism, and that includes synonymous mutations. Now, if a substitution is not synonymous, then it means it's non-synonymous. It's going to have an impact on the amino acids for that coding region. Okay, so non-synonymous mutations come in two versions. A missense mutation is where we have a mistake, a mutation that causes a amino acid to be changed into a different amino acid. So let's go back to our example. We'll stick with the same one. If the parent has the sequence CCC in a particular codon, and now this middle base pair mutates into an A, we would then have a CAC, which is a histidine, a different amino acid than proline. So that's a mutation that would change the DNA. It would also change the amino acid in that protein. So that's why we call it a non-synonymous mutation, because we don't have the same amino acid. Okay, and so non-synonymous mutations that change a single amino acid are a missense mutation. A nonsense mutation is where a codon that usually codes for an amino acid is changed, it mutates, just a single base pair changes, and that changes into a stop codon. So now we're going to have to use a slightly different example. So notice that if we had UGG, that would be tryptophan. So that's one of the types of amino acids. But if a mutation occurred, a mistake was made, and we now had UGA, we would have a stop codon. So that would be a missense mutation. Now, when we look at the phenotypic impact of these three types of substitution mutations, a synonymous mutation and then the two types of non-synonymous mutations, we can generalize them and put them into classes of phenotypic impact. A synonymous mutation has no phenotypic impact. We changed the DNA, we did not change the amino acid. Okay, and by definition, synonymous and silent mutations have no phenotypic impact. A non-synonymous mutation, so a missense mutation, there's a wide range of impact. Sometimes when we swap out one amino acid for another amino acid, which is a missense, then we have very little or even no phenotypic impact at all. It's a real change in the DNA, it's a real change in the amino acid. But if we swap out an amino acid for another very similar amino acid, that protein might work just the same way it did before. And so we could have essentially a silent mutation even if it was a missense mutation. Now that's not very common because most amino acids do make some sort of a phenotypic impact on the gene that they're in. And so if we swapped one out for another one, we could have a very severe impact. So it can be range from almost nothing to all the way to very severe, even fatal. A nonsense mutation is almost always a very severe mutation. So for instance, if we were to change an amino acid in the middle of a gene to a stop codon, that would truncate that gene. It would be half the length it was before, and almost certainly would make it non-functional. And even cutting off a gene near the very end of the sequence, you're still going to have some sort of usually very severe phenotypic impact. So most nonsense mutations have quite a large phenotypic impact on the organism that carries them. Okay? So an insertion deletion mutation is another type of a point mutation. We're going to change one base pair. But this is when we insert or delete a single nucleic acid from the DNA sequence. And these also usually have very large phenotypic impacts. The reason for that is because we change the reading frame. So in the genetic language, unlike the English language, there are no spaces between the codons. So there's no way to determine what the correct reading frame is just by looking at the DNA. So we have to use other methodologies. And in the worksheet, you'll look at some of the ways that people look at long stretches of DNA and determine the reading frame. So if you just have a long stretch of A, C, G, 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 T, A, C, C, whatever combination it is, you don't know the correct reading frame until you've done a little bit of additional work. And if you took out one of those nucleotides, or if you inserted an extra one somewhere in the middle, that would change the reading frame for everything downstream. Now, today I will post in this same folder, so in Tuesday, July 23rd, I will post a challenge. It's going to be for an attendance grade. It will pop up at midnight on July 22nd, and will be there all day throughout July 23rd. It will disappear at midnight on July 23rd. So you need to complete it sometime in that 24-hour period. I posted that. I announced it in class. I posted it on the announcements on Blackboard. I'm telling you here again, reminding you. So hopefully you're looking at this on the 23rd or before, and you'll remember, you'll do that little challenge. You'll get an attendance grade for it. Now, in that challenge, we will do an analogy to the English language and review and record some of these possibilities for synonymous, non-synonymous, and then show you the impact of an insertion deletion. And we're going to use the English language as an analogy for the genetic language. It's not perfect, but it works well enough. So please make sure you do that, and you'll get a little bit more of an overview of these point mutations. Now, there are other types of mutations that can occur, and most of the time these large mutations have a very large phenotypic impact. But some of these large mutations can be effectively silent. They can have no impact at all on the phenotype. So realize this question down here does not have a simple single answer. There's going to be a range of phenotypic impacts, although generally, if you had to bet, it would probably be fairly severe. So in a way, it's kind of like asking, what effect does a car accident have on an individual? And realize that usually it's going to be severe. At least it's going to shake you up or worry you. Maybe you don't get physically injured, but many times you're going to have physical damage to your car, maybe even damage to yourself. So there's a wide range. We need to look at each each type of mutation and evaluate it in its context to figure out what the phenotypic impact was. Okay, so here are 4 types of large mutations. First is a duplication. Now this is a duplication of a large area of a chromosome. So over here you have an example in letter A. We've got a chromosome with 8 different genes on it. Sorry, just 7 different genes on it. And by mistake, 2 of those genes were copied twice. And so now we have a chromosome with extra genetic information. And so duplication type mutations mean that you've got redundancy at the level of genes. You have entire genes that are now represented twice. And again, usually this is a fairly severe phenotypic impact, although in some organisms, carrying a few extra genes would have very little or maybe even no impact at all. It's going to depend on the genes and the organism that we're looking at. The second type of large mutation is a deletion. And a deletion occurs when we are missing part of a chromosome. Now, sometimes duplication and deletion mutations happen together. So remember how we talked about crossing over during meiosis, when a chromosome swaps parts with its sister chromosome? And most of the time when that swapping occurs, they swap equal parts. And so the chromosomes don't end up any larger or smaller. We're not missing any material or having extra material. They just have maybe a different combination of alleles. But sometimes when that crossing over occurs, it's an unequal crossing over. And so in the analogy we used of looking at your partner and saying, hey, you've got a nice arm. Hey, I like your arm. Let's swap. You swap arms evenly. Everyone still has 2 arms and all the parts there, although there's going to be more variety and diversity. But now imagine that you looked at your partner and said, hey, I really like your hand. And your partner says, yeah, but it's a really good hand. I'll give it to you, but only if you give me your whole arm. And so your partner takes off their hand and gives it to you, and you take off your entire arm and give that to them. So now they would have extra material, right? And if this is a chromosome, you'd have extra genes. And then the other one would be missing some of the genes because they did an uneven swap. And so sometimes these duplication and deletion mutations can happen side by side, and one gamete would be carrying extra genetic material, and the other gamete would be carrying less genetic material. Okay, but there are also versions of them that can happen that don't have kind of this equal effect on either side. You can just get extra material if you copy too much, copy more than you needed to. Okay. Now, lastly, 2 more types of larger mutations. We have an inversion, and an inversion occurs when we flip around part of a chromosome. So here they've written them in like a mirror image script here backwards. But you have 7 chromosomes. You still have 7 chromosomes here at the bottom after the mutation, but they're now in a different order. And again, these can have a variety of phenotypic impacts. Some inversion mutations have no phenotypic impact at all. The genes are still there, they can still be used. Other times their ability to be used is affected by that inversion. And then finally, a translocation. A translocation is where we move material from one chromosome to another part of that same chromosome or to a completely different chromosome like we've done here. So notice that we've moved the Q and the R gene onto this chromosome that has the A through G genes, and so now we have two very different chromosomes. And so in a way, it's not quite like an insertion deletion mutation because we still have that material. It's still there. It's just located in a different place. So we call it a translocation mutation. And both inversion and translocation mutations happen rather frequently, and they cause genomes to be rearranged. And so when we look at genome evolution, we'll kind of talk about that a little bit and talk about the impact that they have on genomes. I mentioned this in the little review overview of DNA code and mutations that is also posted in the same folder. But you should realize that essentially mutations are random events. They can then be propagated and passed down to offspring. So if it's a very recent mutation and hasn't gone through many generations of reproduction, it's not going to be common in the population. Occasionally there can be convergent mutations, so individuals can end up looking similar despite not being descended from the same mutated individual. To determine that, we would need to do a phylogeny. Okay, and so we'd need to figure out whether it's independent acquisition of the same trait or whether it happened once and all of the organisms have inherited that from their common ancestor. But essentially, mutations are random. We are going to look at them as if they are small mistakes, sometimes large, but mistakes that happen during DNA replication, and they could just as easily happen in one chromosome as in the other part of the chromosome. And after they occur, then the processes of genetic drift and natural selection can work on them. Okay? And mutation rates vary quite widely between organisms. Bacteria and especially viruses tend on average to have a little bit of a higher mutation rate, and that works really well for them. Eukaryotic organisms tend to have lower mutation rates. Now, often people think of mutation and there's kind of this bad connotation of what a mutation means. But realize that mutation in and of itself is not a good or a bad thing. We would need to evaluate each individual mutation and determine whether it is good, bad, or neutral. Most mutations, when they occur, are neutral. We're going to look at the reasons for why that is in a coming unit here. But just know for now that most, maybe as many as 95% of all mutations are neutral. Of the remaining ones, those remaining 5%, the vast majority of those mutations, maybe 4.9999%, are detrimental. They're bad. The organisms that carry them do not do as well as if they didn't have that mutation. And so natural selection works to remove those negative or detrimental mutations from the population. Over time, they get removed. And then the remaining 0.0001% are beneficial. So having a beneficial mutation is often a very, very rare event. But when it does happen, it gives an advantage to that organism. They do better, have more offspring, and soon the vast majority or every individual in that population carries that new mutation. And so they become common, and that's what we look at, what we observe in natural populations. Okay, so next unit we're going to talk about genomes, and what we see is we've been able to evaluate the genomes of organisms. Again, reminder, you need to, in this same folder, in the Tuesday, July 23rd folder, you also need to complete the challenge, which will give you an attendance grade for today. So that's like if you were in class in there and turned in one of the sheets. And in that challenge, we'll review the genetic code. We'll use the English language as an analogy for that genetic code. So please remember to complete that. And again, if you have any questions or anything that you need to clear up, feel free to send me an email and I will check those daily.